Hi, I am Ed Pistilli, and I am an associate professor in the Division of Exercise Physiology with a secondary appointment in the Department of Microbiology, Immunology, and Cell Biology, and I am a member of the WVU Cancer Institute. The research in my lab addresses an important problem in clinical oncology that currently has no treatment options available for patients, and that problem is breast cancer-induced muscle fatigue. If left untreated, tumor growth and tumor-directed therapies can significantly impact the functional capacity of patients, and muscle fatigue is an independent predictor of patient survival in a number of different cancer types. So the way that my lab addresses this issue is through basic and translational experiments including the growth of human and mouse breast cancer cells in culture, mice implanted with human breast tumors, and muscle biopsies from breast cancer patients. Through a collaboration with the WVU Cancer Institute and breast surgeons, my lab has acquired over 80 muscle biopsies from patients with breast cancer. And these biopsies have been used for next-generation RNA sequencing and proteomics analysis. And we've been able to generate a global molecular signature of skeletal muscle during breast cancer. The genes and proteins involved in this molecular signature are involved in mitochondrial function and metabolism, and predict a deficiency in ATP availability within skeletal muscle. In addition, the muscles from our mice implanted with human breast tumors show a strikingly similar molecular response pattern. So now we have a mouse model that recapitulates the human condition from a gene perspective, a protein perspective, and a functional perspective. And we're now using this mouse model to conduct preclinical studies using a number of drugs that can target mitochondrial function, ATP deficiency, and muscle fatigue. Students in my lab are exposed to a number of different areas of research, including muscle physiology, gene and protein expression techniques, bioinformatics analysis of large data sets, cell culture, and mouse models. And I challenge my students with the same question I ask myself every day. What have we done today that brings us closer to a therapy for patients experiencing fatigue? I have routine lab meetings with my students each week where we discuss experiments, we analyze data, and we troubleshoot any issues in the lab. And I encourage my students to act independently and follow their ideas. So feel free to contact me if you'd like any more information on the lab or our work, and welcome to WVU.